I'll get right on with this. So I want to explain a couple things first before I get started. First off, I want to explain that what I was charged to do here was to give a public talk about something to do with my research. And so I'm in the unusual position of giving a public talk about a research field who's populated by a number of extremely notable figures who are here in the room. And so I'm going to apologize to them right now. If I make any mistakes in details, maybe we can talk about that later. If I overgeneralize about something, then maybe we can argue about that now, because I'm going to try to give a sort of a general feel for some things that I think are important. The microbial world is a particularly difficult thing to give a general talk to the public about because it's very abstract. And so that's one of the things I want to talk about today, is sort of how to get our heads around the abstract nature of microbial life. The other thing I want to emphasize, coming back to from time to time, has got something to do with an important part of science that I feel is very important, that's often overlooked, which is the importance of exploration. We all sort of acknowledge at some level that basic research is important, and I of course agree, but there's also a level of exploration that's necessary to go out and even find questions. We can't really test hypotheses for many things because we don't even know what the right questions are yet. So to some extent, as I like to sort of think of it, we need to just get in the boat and go look at things and see what we find. And from that, we can formulate some of our questions. And so I want to emphasize that a little bit too, because I personally think that's an undervalued part of research that's going on today but isn't given its due. And so I'm going to use that sort of as a theme to get started here. I've forgotten what title I had. Can you see this, by the way? Is there any laser on there? No, good. Okay, well, we'll deal with that. So I want to talk a little bit about exploration to start with. And I titled my talk, Here Be Dragons, which is a phrase that came from the Lennox globe. It was a medieval cartographer's notation for the unknown. And it's kind of gone into general speech as being sort of the unknown that we don't know anything about. And it's supposed to be scary. It was at the edge of what had been mapped, and it was a scary place where monsters lived. And that's fairly understandable when you get to the edge of the mapped globe, that after that is a little bit frightening. But I want to emphasize something else about it. The monsters that these people drew were not actually very imaginative. If you look at them, on one level you think, yeah, that's a strange monster, but they're really not. They're just recombinations of things that people were familiar with. So you stick a lion's head on a fish or something like that. So, and I want to point this out because we're actually not very good as a species of imagining things that are truly alien life, things that are truly unknown. We're actually not very good at thinking about that. Some science fiction authors are quite good at it, but as a general rule, we're not very imaginative about the unknown. And in case you think this is just something that medieval cartographers dealt with, it's not true. If you go through more modern history, this is taken from the cover of a pulp novel where we have this terrible alien attacking a Martian settlement. And the terrible alien is just a great big bug, basically. It's nothing very imaginative. I always like to bring in Japanese pop culture into this because they have a lot of this kind of thing, and it's also just sort of different reorganizations, recombinations of animals, basically. They're all vertebrates. They're all usually bipods or tetrapods, and in this case a giant lizard that shoots lightning or fire or something like that. Now, where it gets really ugly is when we start to imagine what intelligent aliens might look like, because they basically always end up looking like us, but with big heads. And we're all familiar with this. We've seen it many, many times. I used to like to get them out of the Weekly World News, which is out of business now, which is really too bad, because it was a great source of fantastic imagery. And they have a whole series of these, by the way, where they consistently, successfully called presidential elections, and it was the same alien each time. Anyway, the point, though, is this is about as unimaginative as you can get about imagining alien life forms. So why does this matter to biology, to a biologist? Why should I care about this? Well, if you look at the history of biology, right, from when we first started asking questions about biology till the present, for the vast majority of that history, biodiversity has been very easy to understand because there's only two things. There's animals and there's plants. And we've been very good at telling the difference between animals and plants. we have a very strong intuitive notion of what's a plant and what's an animal. So in this picture, there's an animal, which I can't point to, but you can probably see it, that's masquerading itself as a plant. But if you wanted to know the difference, all you had to do is bend over, pick it up, and flip it over. And you will get a lot more information immediately that will tell you that this is a moth, not a leaf, okay? Because we have a notion of what's an animal and what's a plant that's burned into our head, probably by evolution. So this was, as I said, for the vast majority of the history of biology, a very simple problem. There was this nice, comfortable dichotomy between animal and plant. Now, this should have all come crashing down, I think, in the 1600s with this man, who's Anton van Leeuwenhoek. And he was a draper. He wasn't a scientist. He's actually got a really great story that I'm quite fond of. So Leeuwenhoek was a well-respected merchant in Delft, and he dealt with cloth, so he was a draper. And he had lenses that he used to count thread counts of his cloth. And he became interested in these lenses and in looking at different things with his lenses, and then he started making his own. And he made these wonderful microscopes that were very simple single lens microscopes. And he could see things with them that no one else could see. And he looked at everything. He looked at crystals, he looked at pieces of his own body, he looked at animals, he looked at pond water. And he discovered lots of things. But the thing that he discovered that was the most significant was that life could exist as a single cell. Because up till then, we knew about cells. There were good microscopes. There was a cell theory. But no one guessed that they could exist as a single cell life form. So Leeuwenhoek discovered this. He didn't invent the microscope. He discovered the microbial world. And he sent a letter to the Royal Society in London, which is what you would do then because it was the only scientific journal that existed. And the Royal Society got this letter from a draper in Delft that said that basically, you know, everything we know about biology is on its head because I found these microbial creatures that exist as single cells. And they didn't believe him. I guess realistically, why should they? And so they did what the Royal Society does. I guess that's this one. And they formed an expert panel. And they dispatched it to Delft. So the expert panel consisted of a lawyer and a doctor and a vicar. And they went to Delft and they checked on Leeuwenhoek's work. And here's where the story differs from what you're expecting, because they came back and they said he's right. He has discovered single-celled life. His microscopes are great. So they published his letter. And Leeuwenhoek actually went on to be very widely respected scientist, a gentleman scientist in his day. And he published many, many, many letters in the Royal Society's proceedings, which you can all download now through the web, even from the 1600s. And I encourage you to go read them. They're really interesting. But as I said, this was really the seeds of a revolution that Leeuwenhoek sowed. But I would argue that it didn't lead to anything because we didn't know how to handle this information. Scientists didn't. Because what Leeuwenhoek discovered was really a whole universe of alien creatures that didn't fit into our comfortable dichotomy of animals and plants. These things were neither, or some of them were both. There's animals that swam around but also photosynthesized. And people didn't really know what to do with them. So they did what you might expect. For many, many years, they just sort of tried to pigeonhole everything that was discovered about the microbial world into either animals or plants, even though many things patently didn't fit in either. And so this is not ancient history, because to a large extent, this is still affecting the way we think about these things today. Our concept of biodiversity, I would argue, hasn't really changed that much since then. We still have a largely animal-plant-centric view of the diversity of living things on the Earth as a species we do, not me personally. So this is the kind of thing that we think of. We see pictures of rainforests or kelp forests or coral reefs, and we're told this is biodiversity. And what people are looking at are the animals. I actually think that this is a pretty good picture of biodiversity, but the problem is most of the biodiversity is in this you can't see, because mostly it's microbial. But this is our concept of biodiversity. So if you look at, for instance, the UN Red List of endangered species, the last time I checked, there was exactly 0 things on that list that were not an animal or a plant. There might be a fungus on there now, I haven't checked lately. But that is sort of the baseline of our understanding of biodiversity. So does this matter? It does. It affects the way we think about science. So here's here's another example of the same thing. This is a chart that you see repeated a lot about how many species of different kinds of things exist on the planet. And if you add up all the animals, which go from the top of insects all the way around to chordates, it's about 3/4 of the species. So people think this is how many species exist. 3/4 of life on our planet's animal. And then there's a big chunk of fungi and plants and then a bunch of other stuff. And this is a widely reported thing that you see in the David Suzuki Foundation or whatever, and it ultimately comes from the UN. At the scientific level, we see things like this. This is a tree of life that's circulated by a prominent scientific publishing company, Biomed Central. And in it you can see, I've helped out here by circling things. You can see there's a big group at the bottom of animals. There's another group of animals. And we got a couple of fungi who, not surprisingly, are the ones that we eat, mushrooms and baker's yeast. And then, of course, there's lots of plants. And then there's this kind of embarrassing little tacked-on brown smudge called microbes. And it's a single branch, and it's at the base of the tree, and so we have all this baggage about higher organisms and lower organisms coming out here as well. And I'll also contend that it affects how we view time. So if you look through the web for some sort of illustration of time of the geological periods on the planet, it's pretty easy to find these sorts of things. And so at the bottom we have the Precambrian, which is a fairly short phase that's not very well subdivided because we don't know much about it. Most of the rocks are gone. There was life forms there, but they're kind of not very important ones, and we don't know much about them. They're kind of boring little microfossils. And then if you look up around the Jurassic, say, things start to get interesting. I'll just use that as a time point because we know about the Jurassic. There was dinosaurs, you know, they're cool. Dinosaurs are great. The really important stuff is up at the top where we have really well-defined, well-subdivided idea of the geological history of the Earth, and really important things were happening, like us. We evolved. And I tried to find, in case you're wondering, I tried to find bobblehead dolls of Darwin and Wallace. And it turns out to be really easy to find bobblehead dolls of Darwin. But when you try to find one of Wallace, all you get are basketball stars, football stars. And so I included this just to say I'm sorry to Wallace. He's not given the credit that he's due. So the problem with these things is not that they're wrong, it's that they're not drawn to scale. They're not wrong in fact in how they've subdivided things. They're wrong in the scale. They haven't drawn them to scale. So let me show you what I mean by this with the tree. If you were to draw a tree based on, I guess, about as an objective a thing as we can come up with right now, which is molecular data, which is the level at which diversification evolution is happening, you don't get the tree like what the Biomed Central guys have. You get a tree that vaguely looks something like this or some variation of this. But the point is, there are three groups of life, but they're not animals, plants, and fungi. They're bacteria, archaea, and eukaryotes. And within the eukaryotes, we find the animals, plants, and fungi. So most of you probably know this, but eukaryotes are complex cells with their DNA encased in a nucleus, and archaea and bacteria or eubacteria are simpler, structurally simpler cells whose DNA is free in the cytoplasm. So most of the diversity of life on our planet are bacteria and archaea. And within the eukaryotes even, except for the animals, plants, and fungi, and one or two other lineages, it's also all microbial. So in other words, the brown smudge on the first tree represents the vast diversity of life on the planet. Animals, plants, and fungi are important, don't get me wrong. They're very important, but they don't represent biodiversity very well. They're a very small fraction of biodiversity. And if we look at time, we see the same thing, and it actually makes a lot of sense. So here's a depiction of geological time that is drawn to scale. And I apologize, I can't really point, but down near the bottom, there's some text you can't read, and that is when we have our first evidence of cellular life through fossils. At the very bottom, at the very start, is where the Earth's surface cooled, and at the end here is the current, present day. So just for argument's sake, we'll take the Jurassic, which we've mentioned was where the dinosaurs were in that first thing. And let's work our way back and see how far back is that really. And if you work your way back around this coil, you see the Jurassic is actually less than half a rotation from the present. So dinosaurs were a long time ago, by our standards, but in terms of geological history, they weren't actually that long ago. If you go around about one more half rotation, you get to the origin of land plants already. So it also wasn't really that long ago. If you go around a couple more rotations, you get to where the origin of animals. And from that point down, those many, many, many rotations, all the way back into this little speck, it's only microbial life. That's all that existed on the planet. So for most of the history of the planet, there's only been microbes. And so if you think they weren't evolving, you're wrong. Of course they're evolving. So since the dawn of microbial life till the present, they've been evolving. They're changing, they're diversifying. So it stands to reason that most of the diversity on the planet today is still microbial, because it always has been. These two things actually fit together quite well. So where does this come from then, this idea that 3/4 of the life on the planet's animals? Well, there's a trivial answer, which is certainly partly correct, which is that this is what we experience. Our senses have evolved to be able to detect and distinguish animals and plants quite well. And so this is our everyday experience. When you walk outside, you see animals, you see plants, you eat them, you can touch them. They're intuitive things to us, whereas the microbes are not. The only time you can really sense them directly is when they smell bad or make you sick. So that makes sense, and that has a lot to do with it. But just for fun, I looked into another possibility here, which is a related possibility. And I looked into taxonomists. It turns out there's a database where people have self-identified themselves as experts in the taxonomy of a certain group. And if you take these same groups and look at how many taxonomists exist, it turns out that the number of taxa on the planet that we estimate exist corresponds pretty well to the number of taxonomists that work on that group. So I've just colored all the animals blue, and you can see in the number of taxa that we think exists is about 3/4, and the number of taxonomists that work on this group is about 2/3. It's pretty similar. In fact, most of the groups are actually fairly similar. The one glaring exception is the plants, which you can see that the plant taxonomists need to learn to exaggerate more. Because I think they're just being too honest about how many plants they feel exist. So then the next question, whenever I make arguments like this, that taxonomists are just unduly affecting our view of biodiversity, I'm immediately asked, well, how many taxa do exist? And I actually don't really like answering that question because I just don't know, and I don't think anybody knows, and frankly, we don't even know what a species is for a lot of these things. But if I had to take a wild guess, I'd say it might look something like this, where most of the diversity of whatever you might call a taxon is probably viral, because the saying goes, there's about 10 viruses for every species of cellular life. And then within that, I suspect that most life is bacterial and archaeal, just because they've been around probably twice as long. And then within the eukaryotes, the complex cells, animals, plants, and fungi are fairly small wedges, and the microbial eukaryotes, collectively we call the protists, probably represent most of the diversity, because that's what the molecular sequence and the tree tells us. So, as I said, I'm not really slagging this as a view of biodiversity, because there's an awful lot of biodiversity there. I'm just saying it's not a very good view of biodiversity, because most of the diversity in this picture is invisible to us. It's all microbial. The corals, the seaweeds, the fish, they're just a thin veneer on the top of a basically microbial ecosystem. If you were to take a picture of what the biodiversity actually looked like, it mostly looks like this. I like these pictures because they kind of look like a picture of deep space from the Hubble telescope. But what it actually is a little bit of seawater that's been stained with a dye that makes DNA glow. And so what you see is that the seawater is actually chock-a-block with microbial life. There's numbers here that are hard to imagine that we'll get into in a minute. But for now, I want to make a different point, which is that it's not all the same. We have this tendency to say microbes, like it's a black box that's sort of a kind of a bunch of things doing the same thing. Well, they're not the same at all. In this picture, we've got 3 different sized blobs. The big blob up at the top there would be a protist. It's a complex cell like our cells with a nucleus, this one being a diatom. Down on the right-hand side there, there's a sort of medium-sized blob that's probably a bacterium or an archaeum, so an archaeon, so another cellular life form, but one that's prokaryotic. And then there's tiny, tiny little dots that are very hard to see but extremely abundant, which are viruses. So each of these groups, the protists, the bacteria, the archaea, the viruses, the diversity within that group is greater than the diversity, say, between animals and plants. So these represent hugely diverse groups. And the diversity between them isn't even comparable. It's an enormous amount of biological diversity in this microbes black box. We can't treat them all as being sort of a bunch of the same things. They just aren't. Here's a couple just fun numbers that I've collected from colleagues over the years to illustrate some of the importance of these things in terms of bulk on our planet, say. So this is an interesting fact. If you counted up the number of bacteria on the Earth, it's more than there are stars in the known universe. And it's a lot more. It's not just a bit more. It's 6 to 8 orders of magnitude more. So you can sort of picture this in your head a little bit about how many stars we know about. In our bodies, our own bodies, microbes are said to outnumber human cells 10 to 1, which is leading this new idea that really human bodies and all animal bodies are really microbial ecosystems. So you could sort of think of yourself as a bit of a shell for the microbes that are keeping you alive. And then another one about viruses. In a mill of seawater, there's been estimated from 1 to 50 million viruses. So think about that the next time you go swimming. And they kill 20% of the living material in the oceans every day. That's the part that I find interesting. If you think about the gigatons of carbon that these things are turning over in the ocean every day, it's just staggering what kind of effect they're having ecologically. In fact, one of the things I've learned in the last few years hanging out with more ecologists is that all of the important sort of geochemical cycles that are going on in the ocean that affect things like climate, ocean acidification, and whatnot, they're being driven by microbial processes in the ocean. The ocean's one place where the animals and plants really just don't seem to matter very much. So these are big numbers, and the scale of the microbial life is a persistent problem when you're trying to think about it. And I want to talk about scale a little bit, and particularly the size of things. Try to see if we can wrap our heads around what this world's like for a bacterium, say, because it's not the same as it is for you. So I'm going to use a cosmological comparison here for a second. You have to bear with me because it's going to seem totally off topic for a while, and maybe it is, but it's kind of fun to think about. Because it has to do with the cosmological scale and wildebeest. Wildebeest are actually a really fantastic way to think about most problems you have. If you have a problem, I advise you to immediately just think of the wildebeest and how a wildebeest could help you solve that problem. So that's what they're going to do for us today. So we have let's say an ecologist that's interested in the Serengeti and wants to know about wildebeest. And he's in his Jeep here, and he's at a scale where he can study the wildebeest adequately to understand some very basic things. Let's just say, for argument's sake, that he wants to be able to count the wildebeest in the herd. He wants to be able to see what they're eating and to see what's eating them. Real basic stuff, okay? And he can do that. From that position, he can gather that kind of data fairly accurately and understand those simple questions. All right, so now here's what I want you to think about. Think about a microbial ecologist that wants to understand the same sorts of things about a bacterium that lives anywhere, say in the ocean. Now, they've got a problem of scale. The bacteria is so small, it's hard to put yourself in their world to understand how they're interacting with other things. So let's do the opposite and say, how far away would you have to take that wildebeest ecologist from the Serengeti for them to have the same problem of scale? So that they're so far away that the wildebeest are as small to them as the bacteria is to us sitting at our bench, okay? Because this will make it really easy to understand, right? Because the number's 250,000 kilometers. So now you all know, right? Oh, I get that. Now I understand. Because what's 250,000 kilometers? I actually don't understand it because that's such a big number that I don't know what 250,000 kilometers is like. I can't put myself in the wildebeest ecologist's position because that's such a huge number. So I thought to myself, well, how can I illustrate this? And I decided I would see if Sputnik would help. Because I thought, well, maybe Sputnik's about 250,000 kilometers from the Earth, and that would give us some sense of the scale, right? So I went and looked up, well, how far away was Sputnik from the Earth? Because I didn't know this either. And it turns out that that doesn't really help at all. Because Sputnik was only 250 kilometers from the Earth. All right? So then I thought, well, surely the space shuttle goes a lot further away than Sputnik. It does, but it only went about 1,000 kilometers away from the Earth at the most. And then I had a really dumb idea, and I thought, well, what about those space stations? But I wasn't thinking the fact that, well, if the space shuttle didn't go as high as Mir, then that's a problem, right? That's fairly basic. And of course, that means that ISS, even Skylab, wasn't really that far from Earth, if you remember Skylab. So that didn't help. So then I thought, well, gosh, the moon is a long way away. Although it's getting so far away, it's not even helping much because it's kind of abstract. So how far away is the moon from the Earth? Would the wildebeest ecologist have to be on the moon? Turns out not quite. Not quite to the moon. This is almost drawn to scale, and this is the Earth and the moon. And the wildebeest ecologist would have to be about 3/4 of the way to the moon. Okay? So think about that for a sec, because actually, I believe when you really think about this, it does help. So if you imagine yourself on this wildebeest research station orbiting 3/4 of the way to the moon, and you're supposed to be able to tell how many wildebeest are on that piece of the Serengeti, what they're eating, and who's eating them. Even if you have the best telescope available from the NSA, they probably have the best one, I don't think you're going to be able to do it. In fact, I think you're going to have a hard time telling a wildebeest from a blue whale. And what's more to the point, I think you're going to have a really hard time finding a lion, say, because they're rare. And these are all problems that we do have in the microbial world. Basically, when you get right down to it, even though we're not very good at thinking about alien life forms, the scale of the microbial world is so extreme that it's not even really a matter of scale anymore. It's just different to the point where it is an alien world. There's things in the microbial world that matter a lot that we don't care about, like viscosity of water, say. But there's things that are really affecting us that don't affect most microbes. Like many microbes don't care that much about gravity. Some do, some don't. So it is a different kind of world that they live in that's really hard for us to put ourselves into. And so we've been well trained through Hollywood or TV or comic books to know that to get into such a place, to move to an alien world, you need some sort of secret door, like a portal, right? And we can't transport ourselves physically into the microbial world, but again, I would say that Leeuwenhoek actually presented one of the two ways that we can get into that. The first that I'm not going to discuss much is microscopy. Microscopy has changed so much, and it's changing right now so much, that we can do things today with microscopy that we would have considered to be pretty much magical when I was doing my PhD. And so I actually think the future of this field will be a lot around imaging and microscopy. But what I'm going to talk about today is more the second, which is interestingly also even more abstract than the microbial world itself, I think, which is molecular sequence data. And that's because molecular sequence data is what gave us the first clue into just how big this problem really was. And it's the first way that we've been able to tackle it. And so that's what I want to discuss more today. So I've picked a particular instance from my own research that I wanted to use to illustrate this. And I'm going to try to do so in fairly broad terms, nothing too specific. And I like it because it brings together a couple of things that I think are important. The first is just the vastness of what we don't know about the microbial world, because there's an amazing depth to our ignorance that's good to acknowledge. But the second is the importance of exploration, or basically chance. It's Without chance discoveries, we would be spinning our wheels, and these come through exploration. So I'll tell you a little bit about this. And it brings together two things that we wouldn't have put together not very long ago. One is the origin of parasites like the one that causes malaria, and the other is coral reefs. So malaria, just in case anyone doesn't know, is a disease-causing protozoan called Plasmodium, and it infects red blood cells of humans and many other animals, and it also infects other things like livers, and malaria goes through a life cycle that includes insects. It's a terrible disease that kills about 1 million people a year, and is currently treatable but not curable, I guess I would say. Now, malaria is particularly special because what it does is it gets inside our cells. It's an intracellular parasite. And this is a picture of some healthy red blood cells around the outside, and on the inside are 2 infected red blood cells that have malaria parasites. The one's been ripped open, and you can see the parasites inside it. So that's one way that it avoids our immune system, but it's got a very sophisticated mechanism for getting inside the cells of animals. So this organism lives in the dark. So it was a bit of a surprise about 15 years ago to find that it has something called a chloroplast. Now, a chloroplast is the subcellular compartment of some eukaryotic cells where photosynthesis takes place. So in plants and algae, photosynthesis takes place inside this thing called a chloroplast. And this is a toxoplasma, a kind of parasite related to the malaria parasite, and it's got these green dots that are lit up with a fluorescent dye, and that's this thing's chloroplast. So why would an intracellular parasite that only exists by getting inside the cells of animals and infecting them, usually causing disease, contain the structure that plants and algae use for photosynthesis? And where did it come from? These are the two questions that immediately arose from this discovery. Now, the function of the chloroplast, or plastid, was actually fairly easy to figure out. And it was discovered fairly rapidly that there's this set of biochemical pathways that I'm not going to discuss any longer today were discovered to be the reason why this structure still existed in these plastids. So these biochemical functions were taking place inside the chloroplast of the malaria parasite. I'm going to talk about this functional aspect more tomorrow in a seminar elsewhere. Ford's fallen asleep. Okay. But what I want to talk about today is the origin of the structure. So where did the chloroplast come from? Now, before I get into that in much detail, just to make sure we're all on the same page, I want to make it clear that chloroplasts are special structures because they arose through a process called endosymbiosis. So this is where one cell eats another one and retains it, make the two form sort of fuse together and become one cell. So up at the top we have a photosynthetic cyanobacteria in a cartoon form being eaten by a eukaryote, and instead of digesting it, it just keeps it and it becomes part of the cell, the chloroplast. And this process gave rise to 3 kinds of algae: glaucophytes, red algae, and green algae went on to give rise to land plants. So that's a very important event in evolutionary history of eukaryotes. But it does not explain all the diversity of algae, because most algal groups originated by a process called secondary symbiosis, where one of these eukaryotic algae was itself eaten by another eukaryote, either a red alga or a green alga was eaten by some other eukaryote, and went on to give rise to new algal groups, okay? And so it's like cells within cells within cells, like Matryoshka dolls, all right? Now, the plastid, or the chloroplast of the malaria parasite and its relatives, were known to be derived from the secondary symbiosis event, okay? But nobody could tell which kind of algae it was. Was it a green algae or was it a red algae? And this is actually pretty important, because if you want to understand the evolution of the things, you need to know this sort of thing. And it also has some impacts on their function and structure. So, for many years, there was a long debate about this. And I've shown a couple pictures up at the top there where people said that it was a green algae based on some evidence or other. And if you've got sharp eyes, you can see that these are well-respected and high-impact fonts. Okay? Now, down at the bottom, there's a couple of papers that said it was a red alga based on different kind of evidence. And you can see there it's a bit of a mix between well-respected and high-impact fonts and more specialist fonts, okay? So this debate went on and on and on. And for an evolutionary biologist, this is odd, because this question should have been fairly simple. The reason why it should have been simple is what I'll show you here. Apicomplexa at the top include the malaria parasite, that's the name of the whole group of parasites, are closely related to something called dinoflagellates. Dinoflagellates are well known as algae, about half of them are, and they have a chloroplast that's known to be derived from a red alga by secondary symbiosis. So we have a situation where we have two related, glad that wasn't mine. We have two related groups of eukaryotes that have the same structure, and we should just be able to compare them and say whether they trace back to the common ancestor or not. This is about as easy as it should get in this kind of comparative evolutionary thing. But it doesn't work that way because they're both very strange. The apicomplexan plastid you might expect to be weird because this thing's not photosynthetic anymore. So it lost a bunch of genes and it's changed functions and so forth. So that's maybe not that surprising. The dinoflagellates, on the other hand, don't have any particular excuse, but they're about as weird as you get. And they're just weird, I think because they just like to be weird. In this case, they're still photosynthetic, but they've changed their plastid, if anything, even more for different reasons that I won't get into. But the problem is they've changed them differently from each other. So they've diverged from each other, or they arose from different ancestors, and we can't tell which. To illustrate what I mean, these are Venn diagrams of the sum total of genes we've ever found in the genome of that relic cyanobacteria, the chloroplast, because they retain their own genome. And what you can see at the top is all the apicomplexan genes, and at the bottom all the dinoflagellate genes circled in green. And the intersection is very, very small. In other words, they don't even have the same genes mostly, and so we can't compare them. So this is extremely frustrating for an evolutionary biologist to have two homologous somehow structures in related organisms that we can't directly compare. It's extremely frustrating. And that's why this debate went on for 10 years, is because we were using indirect data or proxy kinds of data to try to address this question. Now, the solution to the question didn't come from that. It came from somewhere totally unexpected, and it came from exploration. A group in Australia, led by Dee Carter, was interested in the dinoflagellates that are symbionts of corals. They're photosynthetic symbionts that give the corals their beautiful colors and also give them photosynthate. And so coral bleaching, for example, the disease that's killing dinoflagellates due to global warming, is what happens when they expel their dinoflagellates, and then the corals eventually die. So these are important symbiosis, and important symbiotic relationships. They were looking at the diversity of symbionts that they could find on the Great Barrier Reef. And these symbionts aren't very pretty. They're little brown balls. They're hard to tell apart. And they were using molecular data to tell which kinds were which. And what they found was just one symbiont was special because it wasn't actually a dinoflagellate at all. It looked like it. It was a little brown photosynthetic ball that seemed to be a symbiont of corals, but it was different. It's called Chromera. There's a picture of it. And Chromera is actually related to the apicomplexans, not to the dinoflagellates. In other words, it's kind of like a missing link. I don't like that term, but I don't know what else to call these things, because it's the relative of the apicomplexan parasites, but one that retains photosynthesis that they have lost. And so our hope would be that something like this, if you looked at its plastid genome, might prove to hold the information that both the apicomplexans and dinoflagellates have lost, to bring them all together. So that's what we did. We sequenced the chloroplast genome of this organism, hoping to solve this problem, the non-intersecting nature of the data that exists in their genomes. And what we found is it did quite well. In fact, I should mention there's two lineages. We also identified a second lineage of photosynthetic apicomplexans that existed in our culture collections but had been misidentified as something else. Sequenced both their plastid genomes to tackle this problem. It did so almost beyond our wildest expectations. So if we look at the Venn diagram now, we see that both the apicomplexans and dinoflagellates collectively are intersected completely by Chromera and Vitrella, which means that the dinoflagellates and apicomplexans both evolved from a common ancestor by gene loss. They just lost different genes, which gave rise to this condition where we just couldn't compare them. Moreover, some of the genes have gray boxes around them. Those genes are only found in red algal chloroplasts. They're never found in green algal chloroplasts. And so they also answer the red versus green part of the debate. So now we can say quite clearly that the apicomplexan plastid is related to the dinoflagellate plastid and was acquired from a red alga. So a 10-year debate that had a lot of effort go into it is just put to rest immediately, thanks to a little bit of basic exploration by some colleagues on the Great Barrier Reef. And I should add that phylogenetic analyses and other things back this up. So in other words, we have our answer. And we can put apicomplexans in an even broader context that is actually fairly contentious still, but we can say quite clearly that it's derived from an event where a red alga was swallowed by a non-photosynthetic eukaryote that actually gave rise to quite a diversity of algae. So apicomplexans are surrounded by many, many, many photosynthetic relatives. Exactly how many people are still arguing about. And I don't actually want to talk about that much more, because I want to talk about something else that's really interesting that gave rise to this. It sort of illustrates how this goes in cycles. So this is a picture of a typical molecular ecology experiment where people were interested in bacterial diversity. So what they do is they go out to an environment and they sequence they pick some gene and sequence it from all the bacteria in the environment to sort of say what lives there and try to make some guesses at who they're related to and how they interact. And in red there's something called novel bacterium. And this is a fairly common thing to find, which is a new group of bacteria that doesn't seem to be related to any known bacteria that were only found in this environment. This particular environment was a coral reef. And this new bacterial phylum was just one of many, and nobody paid much attention to it. After we sequenced the chloroplast genomes of the coral symbionts, however, what we realized was they're not bacteria at all. They're actually the plastid or chloroplast of a photosynthetic apicomplexan. But because this experiment had been done before we sequenced the genome, they couldn't possibly have known this. So it got us wondering, there's scores of millions of bacterial sequences out there from microbial ecology, and if some of them are actually from parts of cells of eukaryotes derived from endosymbiosis, maybe we can say a lot about how those eukaryotes are distributed on the planet without doing any field work, say, just to get a good idea. So we did this. We decided to tackle it from a fairly small scale, so we stuck to only the really longest sequences with the most information. And we found 1.6 million bacterial sequences that satisfied our long criteria. They came from thousands of environments, loads and loads of different samples. And we just said to ourselves, how many of these are actually organelles of eukaryotes? And it turns out that after a lot of work, 10,000 of them were, which is actually a lot of data for a protostologist. So then we thought, well, where are the 10,000, what's interesting about these? The first thing we want to know is how many of these chloroplasts out of 10,000 do not fall within an already known group of algae? And it turns out not very many, which tells us that phycologists, people that study algae, have actually been very, very good at identifying the major groups of algae. There aren't hundreds of new groups of algae out there yet to be discovered, we don't think. But there were 450 that didn't fall within a known group of algae. And they shared some very fascinating things in common with each other. The first was the environment. They all came from coral reefs. So if there's a hotbed of algal diversity that we don't understand, it's coral reefs. Okay? The second thing is they all were phylogenetically related. So here's a fairly complicated tree, so I'll go from the bottom up and just explain it. This is a tree that represents all chloroplast diversity. So at the bottom we have the cyanobacteria, which are the kind of bacteria that chloroplasts are derived from through endosymbiosis. And then we have all the major groups of chloroplasts. So the Viridiplantae is where we'd find the plants and the green algae, say. And the numbers beside them reflect how many of those 10,000 fall into that group. And so not surprisingly, the green algae are very common and abundant, and a lot of our samples were green algal. And the same up at the top, the Stromenopiles includes the diatoms, which are also very abundant. We found lots of them. So the data actually sort of reflected what was already known about algal abundance. Now at the very top is where things get strange, because we have the apicomplexan parasites, including Plasmodium, and then this huge group of unknown things. And these are the 450 sequences that don't go with any known group of algae. And they all are related to the apicomplexan parasites. Just to put this into a bit of perspective for you, If you looked at the sequence diversity that these things encompass, it's about the same as plants. So it's not a tiny little unimportant group of biodiversity. It's actually a fairly big group. It's about like saying, well, we missed plants there, we just found them. It's, you know, inconceivable, but it's not inconceivable in the protist world. If we look at the ones that we know about, we have our friends Vitrella and Chromera fall into this group, but they're both parts of big diverse groups. They're not just one species, they're actually just the one that we've got in culture. And the most abundant one, interestingly, at the top is just called apicomplexan-related lineage 5. We don't know anything about it. It's just a sequence. But this is a very important sequence because this is, as I said, the most abundant of the apicomplexans that are photosynthetic in nature. And it's also the most closely related to the parasitic apicomplexans. So actually, if we want to use these things to understand the origin of the parasites, these are the most important ones to study, which is unfortunate because right now, we don't know anything about them. They're just a gray box in this picture. But nevertheless, we have a new way to look at the evolution of these things. This is the tree that I showed you before, but adding this ARL5 lineage just to emphasize that it, in particular, is very important. And now we have the situation where we have multiple independent branches leading up to the parasites, and they're all photosynthetic things that are strictly found on coral reefs. So this is what I said before. We never would have really thought that coral reefs played a role in the origin of these parasites, but now I think they probably did. So let me give you an example. If you can imagine a coral forming a symbiotic relationship with an alga, much like modern corals do with dinoflagellates. It wouldn't be modern corals, though, because modern scalarian corals are actually not that old. They arose fairly recently after the great dying extinction. Before that, though, there was other kinds of corals, like this tabulose coral, which is now extinct, is a fossil coral, or the rugose corals, other kinds of corals that once dominated as reef-building corals in the ocean. There's evidence that they, too, form photosynthetic relationships with other things, with some kinds of algae, but we don't know what kind, and it's probable that the ones that the dinoflagellates now form an association with had not yet evolved. So our thinking is that they probably formed a photosynthetic relationship with the ancestor of apicomplexans. And this is an interesting thought, because the way that the apicomplexans get inside their host cells, generally ultimately to kill them, is a very complex protein and membrane structure at the tip of the cell called the apical complex. And these symbionts that we have in culture have the same complex. And we think they probably use it to get inside their coral hosts, but not to kill it, but in a beneficial symbiotic relationship based on photosynthesis. So one can sort of imagine a story where this system evolved for who knows what reason. It was probably very complex. But in these organisms was initially used in a symbiotic relationship that was of mutual benefit. And it was probably perfected along the same lines as a way to get inside animal cells. Now the corals that these things probably evolved to form associations with went extinct, which doesn't give the symbionts much choice. When your host goes extinct and you're a highly specialized symbiont, you generally probably also go extinct. The ones that would survive are the ones that had some different lifestyle or had formed a symbiotic relationship with something else, like the few maybe that we still find today in modern scalarian corals. The other thing to think about is what would happen if you had a relationship like this based on photosynthesis and something happened that disrupted the balance of the relationship. The most obvious being the loss of photosynthesis, which is a complicated system which gets lost fairly often in evolutionary time. And if that happened and you were using a very sophisticated and efficient system to get inside your animal host and you lost photosynthesis, really you don't have many ways to survive other than turning into a parasite. And it's not much different. If you imagine the balance of the relationship between a photosynthetic symbiont and its host and an intracellular parasite and its host, it's basically just that the balance has been tipped a bit and you're no longer providing much useful to the relationship. So the system might have evolved for good, but it was eventually used for evil. So these are the new hypotheses that we're now pursuing, and ones that I have to say would never, ever have entered anyone's mind had not it been for a little bit of exploration that led to this discovery of these photosynthetic coral symbionts, and also which lead to the next step, which is more exploration. Because the only thing left to do right now is go back to the ocean and find these organisms that we only have sequences for. Arrow 5 has emerged as an extremely important thing in my preferred question of figuring out where apicomplexans came from. And so we need to go out to the coral reefs. We go to Curacao or the Great Barrier Reef or whatever, because this organism is extremely important to us, and we need to get it into culture so that we can sequence its genome, we can look at its interactions with coral, we can do microscopy to see how it's complex, built to get inside coral cells, because we do have some evidence that it probably is a coral symbiont. So this is, I want to just reiterate the cyclic nature of this. The exploration led to the question, not the other way around. And the question leads to the need for more exploration. So I want to just emphasize the need for both of those things. And I think I'm done. Yes, so just to finish, I want to thank, first of all, my lab, which are wonderful and have been consistently wonderful for however many years it's been, many years. And in this particular picture, second from the right is Jan Junoškovic, who's a Czech PhD student who really did pretty much all the work on the photosynthetic apicomplexans that I just talked about, and in collaboration with several other people in the lab, but he's been really a force behind this and changed the way that the questions that we're asking in my lab. Also, the Royal Society, obviously, for organizing the talk. I want to thank them again. And then I want to draw particular attention to CIFAR for supporting microbial diversity research at a very basic level in the form of research networks. The Tula Foundation for sort of, I would say, transforming our field with very generous support for microbial diversity research at UBC and here at Dalhousie as well. And the Guggenheim Foundation, who are kind enough to help me go on sabbatical and try to become a coral reef microbial ecologist with some field work and visits to my colleagues in that field. I'll just stop there. Thanks.